All right, everybody. So this week we're going over plant reproduction. Specifically, we're going to talk about reproduction in gymnosperms and angiosperms. With gymnosperms and angiosperms, they reproduce in similar ways. There are slight differences between the two, and I will talk about what those differences are. And we're mostly going to use angiosperms as an example because they're slightly more complex than gymnosperms. So the idea is if you can understand angiosperms, then you probably understand the reproduction in gymnosperms as well. All right, but do remember, we are talking about seeded plants here. So this is the goal. We are going to undergo reproduction to create a zygote, which is going to be housed in a seed, which can easily be dispersed and help the plant start growing somewhere else. And also remember the difference between gymnosperms and angiosperms. Yes, both of these create seeds. However, angiosperms also create fruits and flowers. So how they reproduce or how they pollinate is going to be different. So with gymnosperms pollination is reliant on wind. So wind is going to carry pollen to an ovule to create a seed. And with angiosperms, pollination is going to happen with the pollinator, whether that be an animal or an insect. Okay, let's talk about some benefits of seeds. So seeds have a seed coat around protecting the embryo and the nutrients inside the seed. So this allows the seed to survive in dry or cold environments. And so the offspring plant can stay dormant until conditions are right for it to start germinating and growing. Another benefit of seeds is that you don't need your embryo to be dependent on the parent plant for nutrition or protection. So instead, the embryo will get all its nutrients from what is stored in the seed. And usually there's enough energy stored in the seed to allow the plant to start growing wherever it lands until it can eventually obtain nutrients for itself with a root system and a shoot system. On top of that, seeds are also very easily dispersed. They can be eaten and carried away, or they can be flung off into the environment. But this allows the offspring plant to grow separate from the parent plant, so there's no competition between parent and offspring. All right, now we need to talk about some of the benefits or adaptations of seeded plants. Now I'm going to compare these to non-seeded tracheophytes and the non-tracheophytes that we discussed in previous classes. All right, you might remember, especially in the non-tracheophytes or the more ancient lineages of plants, that out of the alternation generations, gametophytes were the more dominant stage. While the sporophytes were really reduced and usually needed the gametophyte for nutrition. Now I'm going to be talking about the alternation generations a lot in this video, so if you don't remember your alternation generation, I suggest you pause the video here and go back and take a look at that material again. Alright, something else that we see with the seeded tracheophytes is that they have two different types of spores. They have megaspores and microspores. The megaspore is going to give rise to the female gametophyte, while the microspore is going to give rise to the male gametophyte. So we have this separation of gametophytes at the spore stage. And eventually the female gametophyte will produce an egg, while the male gametophyte will produce some sort of sperm-like cell. We also are going to see the adaptation of ovules now in seeded plants. An ovule is a specialized organ that produces an egg. So you're going to find the tissue that produces an egg within this defined structure called an ovule. And ovules are what turn into the seed after fertilization. So the outer tissue of the ovule will harden into the seed coat. All right, another adaptation we see in seeded plants is that we don't need water anymore for fertilization. With the non-tracheophytes and the seedless tracheophytes, they required water for fertilization to occur. And that's because we had a sperm that was flagellated that needed to swim through water in order to find an egg. Well, that doesn't happen anymore. Instead, we're going to rely on pollen to transport that sperm cell, and pollen is the male gametophyte of your alternation generations. So instead of having a sperm swim through water, now we're going to have an entire male gametophyte travel either through wind or through a pollinator to find an egg where it will then release its sperm cell. And the ultimate outcome is that after fertilization, you have a seed from that fertilized ovule, or the egg inside the ovule. And this is really beneficial because of the things that we just talked about in the previous slide, where you have extra protection and can be transported away from the parent plant. All right, so let's talk a little bit more about heterospory. Now, if you remember your alternation generations, you have a sporophyte that's going to produce spores through meiosis, and these spores with the non-tracheophytes and seedless tracheophytes will be sent off into the environment to grow into gametophytes. Well, we don't really spread our spores with seeded plants. They actually stay on the parent sporophyte. However, the spores that are produced make either the female gametophyte or the male gametophyte, and we can tell this from the spore stage. So this is where these prefixes are coming from the spore stage. With a megaspore, mega means bigger. So these bigger spores that the seeded plants can produce will become the megagametophyte or the female gametophyte. Well, micro means male. So the smaller spores that are made on the parent sporophyte are going to turn into the microgametophyte or the male gametophyte. Now these spores are made from specialized tissue on the sporophyte called megasporangia or microsporangia. And sporangia is simply the tissue that will undergo meiosis to create spores. So you can see that we're using these prefixes over and over again. And it simply translates into whether we're talking about the male reproduction or the female reproduction. So again, we'll have megasporangia that undergoes meiosis to create megaspores. That megaspore will develop into a megagametophyte, which will eventually make an egg. And then we also have microsporangia, which will undergo meiosis to create microspores, which will eventually be turned into microgametophytes, which is the male gametophyte. And that microgametophyte is going to store sperm. And also it's the microgametophyte that is going to travel to find an egg. Now here with the seeded plants, things have switched around in comparison to the non-tracheophytes and the seedless tracheophytes on who is dependent on who. With the non-tracheophytes and the seedless tracheophytes, it was the sporophyte that was dependent on the gametophyte. The sporophyte grew out of the gametophyte somewhere. But that is completely reversed in seeded plants. Here, the gametophyte is completely dependent on the sporophyte. So the sporophyte would be the tree, the parent tree that you would see. That is a diploid structure that is a two-end sporophyte. And through the sporangia will then make the haploid gametophytes. And these haploid gametophytes are going to grow on that parent sporophyte. Now the two gametophytes that are made, the male gametophyte and the female gametophyte, have different jobs to do. The male gametophyte is going to need to be dispersed. So this one-end structure right here that is made out of one-end cells needs to be sent off into the environment in order to find an egg. While the female gametophyte eventually create an egg. So this is a gymnosperm specifically. So in here is an ovule that you'll find on one of the arms of a pine cone. And a pine cone is from Pinus, which is a gymnosperm. So here is your ovule right here. And then inside here, you're going to have your female gametophyte. So this is all one-end tissue in here that is housed by the two-end ovule of the parent sporophyte. And then in here is where you're going to find the tissue that creates an egg. Okay, with gymnosperms and angiosperms, how they undergo pollination is different. So with gymnosperms, they are strictly using wind for pollination. So here's a pine pollen. This is part of the gymnosperm group. And with gymnosperms, you always have these air bladders that are on the sides of this microgametophyte. And these air bladders will help this pollen float through the air to find an egg. That is different than angiosperm pollen. Angiosperm pollen does not have air bladders. Instead, the surface of the pollen has little hooks on them. And these hooks are very specific to the type of pollinator that comes and visits the flower of this angiosperm. So what these hooks will do will then attach this pollen to the pollinator, where they'll be carried off to another flower where they can be released and pollinate that flower. Okay, so we just talked about some of the differences and similarities between gymnosperms and angiosperms as far as reproduction goes. Now we're going to talk about step-by-step reproduction in angiosperms. Specifically with angiosperms, we're going to see flowers being used because remember, angiosperms use pollinators for pollination. And you need to attract those pollinators somehow, so that's what we have the flowers for. We mentioned this in a previous class, but angiosperms also undergo double fertilization, and we're actually going to see the steps of double fertilization now. Part of double fertilization is that you create a zygote, which is going to be the new offspring of the plant, and you create something called an endosperm, which is going to give nutrition to the plant. And something else that we see different than gymnosperms is that we find fruit in angiosperms, and all fruit is something that stores all the seeds that were produced from the ovules through fertilization. Okay, so we have discussed this before. This is the parts of a flower. You should be familiar with these by now. All right, with flowers, you always find both male and female parts to them. So again, this flower is part of the parent plant, which means this is a sporophyte structure, which means this is all 2N tissue. With the flower, you'll find male parts, which are your stamen, and the female parts are usually towards the center, which is your pistil. Now you have different parts to the flower. With the male part, you have an anther, which is going to produce the spores, which again is the microgametophyte. So that is a 1N spore being produced from this 2N anther. And you have a filament that helps hold up the anther to maximize dispersal of this pollen. With the female part, you have 3 structures that you should know. You have a stigma, which is sticky. The stigma is sticky because it's going to catch pollen. Then you have a style, which is a tube that connects this stigma to the ovary. And the ovary is the bottom part of the female pistil. Now you might be asking, what's the difference between an ovary and an ovule? Well, an ovule is that individual reproductive structure that is going to have the tissue to create an egg. While an ovary is something that can have multiple ovules in it. So each one of these ovules will turn into a seed after they're fertilized. Well, the ovary itself is what's going to swell and become a fruit after all the ovules have been fertilized. Okay, so now we're going to go through the steps of double fertilization in angiosperms. And mostly we're going to focus on what happens on the female part of the flower. Now, I encourage everybody to take out a piece of paper and draw this out themselves because that's the best way to learn these steps. All right, so first we need to talk about the development of the ovule itself. All right, so here is my pistil right here that I showed you in the previous slide. So this is all the female parts. So we have our stigma, which is the sticky part up top. We have the style, which is a long tube going down. And then we have our ovules down here in the ovary. So I'm going to focus on just one ovule to show you what's happening here. All right, so let's say this is the one ovule here. All right, first, outside of each of the ovules, we have this tissue right here that we call an integument. And the integument is what's going to turn into that hard seed coat after fertilization. But before fertilization, soft tissue. At the tips of the integument, towards the center of the flower, or this part of the ovule right here, we have an opening right here that we call a micropyle. This micropyle is what's going to allow access of the sperm cell to the egg that's going to develop inside this ovule. Now we're talking about a premature ovule. This ovule is not fully developed yet, and we need to discuss what happens during development. So all this is part of the parent tissue right here. So what ploidy is it? Well, it should be 2N, right? All this is part of the parent, which is a sporophyte, and since sporophytes are 2N, all this tissue must be 2N as well. Well, inside this premature ovule, you're going to find tissue called megasporangium. All right, so this megasporangium is still part of the parent plant, so it's also 2N. And what this megasporangium is going to do is release one cell called, so it's going to release one cell called a megaspore mother cell. This megaspore mother cell was also made through mitosis from the megasporangium, so this cell is also 2N. All right, so the megaspore mother cell is what's going to undergo meiosis to produce a megaspore. All right, so this megaspore mother cell will then divide through meiosis into 4 separate cells. Remember, the product of meiosis is 4 haploid cells. And these cells are now spores, and we call them specifically megaspores since we're talking about the female structure. And these are 1N because they're produced through meiosis. Now, one of these spores are going to turn into the female gametophyte. However, all 4 of them don't grow. Only one of them does. So after meiosis, 3 of these spores are going to deteriorate, and only one of them are actually going to grow to be a megagametophyte. So this one megaspore will then start dividing through mitosis, creating that megagametophyte, that second stage of alternation of generations. Now with angiosperms, the megagametophyte is very reduced. The megagametophyte is actually only 7 cells big, but have 8 nuclei. Alright, so if the megagametophyte is 7 cells and has 8 nuclei, that means one of the cells in this megagametophyte needs to have more than one nucleus. Okay, so I went ahead and drew the cells of the megagametophyte. Now, these cells are existing inside this ovule still. Remember, this ovule is still part of the parent plant, so this is all 2N tissue of the integument. Inside here is the megagametophyte itself. So what ploidy are all these cells? Well, they're all 1N. All right, except for the cell that has 2 nuclei. Now, this cell in the center is called a bipolar cell, and that's because the nuclei are on opposite ends of the cell itself. They're not fused together. So since these 2 nuclei reside in the same cell but are not fused together, this is not considered 2N. This is not a diploid cell. Instead, we consider this N plus N, because it has 2 nuclei that are inside the cell that are both haploid that are not fused together. If these 2 nuclei would fuse together, then we would have a diploid structure, but since we don't, we consider this N plus N. Okay, so now we need to name all 7 of these haploid cells of the female gametophyte inside this ovule. Alright, so the N plus N structure we call a polar nucleated cell. Towards the more superficial side of the ovule, you're going to find 3 other haploid cells. These are called antipodal cells. We're not going to discuss what they do, just know their name. They're antipodal cells, these 3 green ones. Then towards the micropyle of the ovule, that opening, that is where you're going to find synergid cells, which I drew in pink, and the egg itself. Now, these synergid cells have an important job to do. They release chemical cues to help guide the pollen to where there is an available egg. And I'm going to show you that here in a second as we go through the steps of double fertilization. Okay, so before we go any further, I do want to point out that the microgametophyte, or the male gametophyte, which is the pollen, is produced very similarly to the megagametophyte. You are going to have microsporangium that releases a microspore mother cell, which will undergo meiosis to create the microspore. That microspore will go through mitosis to create the pollen itself. However, the pollen does not have this egg or polar nucleated cell. It's all just 1N cells and contains a sperm cell within inside of it. Okay, so let's say this is our pollen right here. There are 2 very important cells inside the pollen that you should know. One is called your generative cell, which acts as the sperm cell, so this is what's going to donate its nucleus. And then you have something called a tube cell. The tube cell is going to grow a tube that connects the pollen to the egg inside the ovule. Now remember, these are all haploid because the pollen itself is your microgametophyte. Okay, so let's say we have our one pollen land on the stigma of a pistil. Alright, the first thing that's going to happen is that tube cell is going to start growing and elongating. And it's going to grow into a giant tube that goes down the style of the pistil and into the ovary, where it's going to find an ovule that has an egg. Now, how did this tube cell find that ovule that has an egg? Because all these ovules might not be developed yet. So how did it find the right one? Well, it's because of these synergid cells that are over here. Remember, these synergid cells are releasing chemical cues to help guide this tube cell to figure out where it needs to go or grow to. Okay, so we have this tube cell now that has grown from the pollen to the ovule and created this very long tube that connects the pollen to the ovule. And so this is the tail end of what's going on right here. All right. So this is that same ovule blown up, and the tube cell found the right ovule because of the synergid cells. That's the only way it knew that this had a megagametophyte inside of it. Okay, after that occurs, the next thing that's going to happen is that the generative cell, which is acting as your sperm, is going to release its DNA down the tube cell. All right, so the DNA is going down the tube cell now. And what ploidy is this nucleus that was released? Well, it is haploid. Remember, this is all part of the microgametophyte, which is also haploid. But something happens on the way down. So as it's going down this tube, it's actually going to undergo another mitosis event. And what that does is create 2 haploid nuclei from that generative cell's DNA. So now we have 2 haploid nuclei instead of just the 1. And this is where double fertilization comes in. So once the nuclei have been divided into 2, one of the nuclei is going to go and fertilize that egg. So now that we fertilized this egg, we have officially created a diploid zygote. So now we have a 2N zygote right here. And that other DNA that was created is going to go past the egg and actually fertilize this polar nucleated cell. Once that happens, all three of these nuclei are going to fuse. All right. So now we have a cell that has three nuclei that are fused together that is actually triploid. And we call this cell the endosperm. Okay, so after fertilization, we have this 2N zygote, which will become the parent plant, the parent sporophyte, and we have this 3N endosperm. What the endosperm is going to do is provide nutrition to the zygote. So that is where the nutrients inside the seed are going to come from, is from this endosperm. We also have other things happen where the integument of the seed, or the ovule, is going to become the seed coat. So this is going to harden and create an outer layer to the seed. And you're going to have that micropyle close. So that micropyle is now going to close to create one solid seed. And then you have a seed that is ready for dispersal. However, with angiosperms, we have this ovary that has multiple ovules in it. So while an ovule will become the seed, each one of these ovules can be fertilized by a different pollen. So you have all these different pollens landing on the stigma and doing the same process, finding an ovule to fertilize, and then an ovule becoming a seed afterwards. Once all the ovules have been fertilized, the ovary itself will then swell to be a fruit. And what you end up with is one fruit that has multiple seeds inside of it. All right, so that's everything for double fertilization. I know it's long, but if you draw this out yourself, it becomes a lot easier to understand. So I would take this step by step. Make sure you understand the difference between a premature ovule, where you just have megasporangium and the megaspore mother cell. And then make sure you understand a mature ovule, where you have the female gametophyte in it, which is made up of 7 cells and 8 nuclei. And then understand what is going on with double fertilization. All right, coming back to the PowerPoint, this is depicting the same thing we just drew. All right, so this is showing you the steps of the ovule development for the female. So real quickly, let's go over this. We have an ovule. Here, it only drew one ovule inside this ovary, but there's usually multiple ovules in an ovary. This ovule is going to have a megasporangium, which will release a megaspore mother cell. That megaspore mother cell undergoes meiosis to create a megaspore. That megaspore will develop to be a female gametophyte, which is 7 cells, 8 nuclei. That is made up of your polar nucleated cell, your antipodal cells, your synergid cells, and your egg. The anther is releasing the microgametophyte, which is the pollen. That pollen is going to stick to a stigma and release 2 cells. The first cell that's going to be released is your tube cell. So that tube cell is going to grow down to find the micropyle in the ovule. And then it's going to release the generative cell, which is your sperm. It's going to release the generative cell's DNA. And that DNA is going to undergo mitosis to create 2 nuclei, where one will fertilize the egg and the other one fertilizes that polar nucleated cell. And what you end up with is a zygote from the fertilized egg and an endosperm from the fertilized polar nucleated cell. And remember, these have different ploidy. The zygote is going to be 2N because it came from the haploid nuclei of the sperm and the haploid nuclei of the egg. Well, the endosperm is going to be 3N because it came from the 2 polar nuclei of the polar nucleated cell and that third haploid nuclei of the sperm cell. And that endosperm is then going to provide nutrients to the zygote as it develops into the young embryo, and the embryo will then continue to use that nutrients until it starts germinating into a new sporophyte plant. So as you can see, the gametophyte stage of seeded plants is very quick. It is the small stage that is found inside the ovule in the female, and the small stage that is the pollen in the male. All right, just looking under a microscope, you can see some of these structures. So here is a cross section of an anther. This is all 2N structure right here, part of the parent plant. And then here you'll find those microgametophytes, which are all 1N. And these are going to have the tube cell and the generative cell inside of them. Again, looking at pollen grains up close from angiosperms, you're going to see that they have hooks all over their surface, and this is because they're going to hook onto the hair of the specific pollinator that they use for pollination. Looking at the cross section of an ovary, you'll be able to see the ovary itself, again all 2N tissue, and then you can see individual ovules inside that ovary. Now these ovules are also 2N, and inside of them you're going to see a megaspore mother cell, since this is a premature ovule, and that megaspore mother cell is also 2N because this is the cell that undergoes meiosis. So this is before meiosis has occurred. And then after meiosis, you end up with 4 megaspores. 3 of them disintegrate, and you're left with 1 that will grow to be the megagametophyte, which is that 7-cell, 8 nuclei structure. And then after double fertilization occurs, you're going to have that ovule become a seed, where that integument of the ovule becomes the seed coat. And then the ovary itself that had all those ovules in it is going to become the fruit. So remember, all fruit comes from the ovary of a flower, and all the seeds inside that fruit come from each of the ovules that were in that ovary. All right, so that's everything for double fertilization. Now we're going to talk about pollination syndrome and exactly how these angiosperms pollinate one another. Now, pollination and fertilization are 2 different processes. Pollination is the process of transporting pollen to another flower. And with angiosperms, the dominant way of doing this is using pollinators, which are either animals or insects. While fertilization is the process of having your sperm cell meet the egg cell to create a zygote. Now remember, this is a huge adaptation for angiosperms and gymnosperms, because with the seedless tracheophytes and the non-tracheophytes, they required water to move sperm to egg. Here, the pollen itself is transported, which is the microgametophyte, to transport that sperm cell inside of it. So here, water is not required anymore for pollination or fertilization to occur. Okay, so now we're going to talk about pollination syndrome. And pollination syndrome is just simply the characteristics of a flower that entice a pollinator to be attracted to it. This is why flowers smell nice and they look pretty, because they're trying to attract certain pollinators to it. So they're taking advantage of pollinators' abilities to see and to smell. All right, so now we're going to talk about different types of pollination in angiosperms. Remember, these flowering plants. And I know I said before that gymnosperms require wind while angiosperms require pollinators, but there are certain angiosperms that also use wind for pollination. Now, this is considered convergent evolution with gymnosperms. And we know it's convergent evolution because these still produce flowers, but their flowers are very reduced. They're not as bright or colorful, and they don't smell as nice. So at some point in evolution, they went backwards or back to wind pollination after having some sort of pollinator in their history. Okay, so some angiosperms require insects for pollination. And again, the angiosperms that require insects are going to take advantage of these insects' ability to see and smell. Now, different flowers are going to require different types of insects for pollination. So they're going to be specialized into the exact type of pollinator they require. And specifically, we're going to focus on bees, butterflies, moths, and flies, and how flowers have adapted to attract these organisms. And one more thing insects are really good at doing is seeing in UV light. So a lot of times these structures that are attracting these insects can't be seen by the human eye. Okay, so one way that flowers can help attract these pollinators or these insects for pollination is using nectar guides. And what nectar guides are doing is creating this target to show the insect exactly where to go to find nectar. All right, so you'll see this a lot in plants. You have these lines that are almost guiding you towards the center of the flower, and that's because that is where you're going to find the stigma to release a lot of the pollen. And remember, insects can see in the normal color spectrum as well as UV light, so sometimes these nectar guides can't be seen by the human eye but can be seen under UV light. Then we find the nectar guides pointing the insect to where the nectar is. Okay, first we'll talk about bees. So bees can see in UV light as well as normal color, and they have an incredible sense of smell. So a lot of times the flowers that attract bees smell nice and have a very strong aroma. Bees also see in UV light, so a lot of times the nectar guides that are on these flowers are going to be in the UV spectrum, guiding the bee to where the center of the flower is, where it can pick up pollen and deposit pollen on a stigma on another flower. Okay, butterflies are another good pollinator. So usually the flowers that require butterflies again have a nice smell to them because butterflies are able to smell, and are usually longer or tube shaped because butterflies use a proboscis to extract nectar. So the deeper the flower gets, the further the butterfly has to stick in its proboscis to get the nectar, which brings the butterfly closer to the flower to make sure it gets pollinated. Okay, so similar to butterflies, we have moths. Moths again can smell, and they have a proboscis. So again, you'll see usually tube-shaped flowers and that smell nice. However, moths are out at a different time of day than butterflies. Moths are usually nocturnal. So typically you'll see these flowers open up during the nighttime instead of during the daytime. And these flowers are going to be in a different color spectrum that helps the moth see it at night. So they're usually more of a pale or dull red, purple, pink, or white, since you don't have the sunlight giving those vibrant colors. And then we have flies. So flies are usually attracted simply by smell. So these flowers are not usually colored or very colorful. However, they have a very strong smell to them. And unlike the other pollinators, flies themselves are not attracted to the sweet scent. They're more attracted to a pungent scent, almost like rotting flesh. So a lot of times these flowers will smell like an animal dying in order to attract the flies. And it's usually more of a trap shape of a flower. So they're trying to attract these flies with a pungent odor, and the fly will then fall into this trap while it gets covered in pollen and eventually escapes with that pollen, and will do the same thing to another flower. Okay, so we talked about insect pollinators. Now we're going to talk about a couple animal pollinators, because there are flowers that specialize with animals. So first we're going to talk about bats. And remember, bats are nocturnal, so these flowers usually open at night. And bats have a very strong sense of smell. So these flowers usually let off some sort of scent. The flowers are more bowl-shaped because now we have a much larger organism that needs to be pollinated. And typically, the odor that is released is more of a strong, musty odor, which attracts the bats. Another animal pollinator that's very common is a hummingbird. Now, these are out during the day, and hummingbirds can see in bright colors and in the UV spectrum. So typically, the colors of these flowers are nice and bright, usually in the red, orange, or white spectrum. Hummingbirds have very long tongues and beaks, so typically these flowers are usually very deep in order to make sure the hummingbird sticks its whole face into the flower, collecting pollen. And since hummingbirds are so reliant on color and vision, these flowers usually don't have an aroma to attract the hummingbirds, and instead just require on their bright coloration. Alright, we've talked about this before with carnivorous plants, and that most carnivorous structures came from flowers. Well, there are some flowers that do both. Use the flower to attract pollinators, but can also eat some of those pollinators. And this is one example of that. So a lot of times water lilies will have these really brightly colored flowers to attract insects. However, these water lilies close at night, so if there's any unfortunate insects still left in the flower when they close, they will be pulled down into the water and drowned. After they drown, they will decompose, and the flower can then use that nutrients from the decomposing insect for nutrition. While a lot of flowers require bright coloration and nectar to attract pollinators, some even try to mimic the insect themselves. This is a specific type of orchid that mimics a wasp, a female wasp to be specific. And what is happening here is that it's trying to attract one of the male wasps in order to mate with it. And when the male wasp tries to mate with it, it will become pollinated and then fly off to do the same thing to another flower. Okay, so that's everything that we're covering this week. So make sure you understand the steps of double fertilization and that you understand what types of characteristics flowers have in order to attract the pollinators that they use.